Imagine looking at, like, a satellite map of a massive battlefield, right? But half the soldiers are completely invisible. Or you can only see one type of uniform at a time. For decades, that is exactly what cancer researchers faced. So welcome to the deep dive. We are unpacking the tumor immune microenvironment, which is basically where immune cells and cancer cells fight it out, and looking at a new protocol called TSA mIHC. Our mission today is taking this highly technical scientific presentation and translating it into a simple, student-friendly guide for you. Okay, let's unpack this.
Yeah, it really is a revolutionary shift. To appreciate this multiplex imaging technique, we first have to look at what scientists call the spatial profiling bottleneck.
Right, because older methods were so frustrating for researchers.
Exactly. Historically, you had two bad options.
Right.
You could grind up the tissue to see all the different proteins, but that completely scrambles the physical layout. Or you keep the tissue intact on a slide, but then you're stuck only tagging one or two immune markers at a time.
So it's basically like knowing Waldo is somewhere in a giant book, but you have no idea who he is actually standing next to on the page.
That is a great way to put it. And what's fascinating here is how TSA mIHC fixes this using an engine of amplification. It comes down to some really brilliant chemistry. So TSA stands for tyramide signal amplification.
Wait, so instead of just attacking a weak tag, it does what?
Well, instead of a weak fluorescent tag, this process uses an enzyme that causes these glowing tyramide molecules to physically bind to the tissue right next to the target. It acts like a chemical chain reaction.
Oh, wow, so it just builds up.
Yeah, it boosts the biological signal 10 to 100 times higher than those older methods.
Okay, but if you want to look at 6 or 7 different cell types, you have to repeat this, right? How do you stop the colors from just turning into a blurry mess?
That is where the denaturation loop comes in. After the first marker is tagged and photographed, the tissue undergoes a strict heat treatment. This heat chemically strips away the initial antibodies, basically wiping the slate clean.
But wait, doesn't the color wash away too?
That's the genius part. The amplified tyramide dye is permanently bonded to the tissue. It stays put. So you can introduce a new antibody for a different cell type without any bleed-through.
Hold on, though. If you are hitting this delicate tissue with severe heat cycles over and over to wipe that slate clean, aren't you risking destroying the very cells you are trying to study?
You are. And that is the exact hurdle researchers had to overcome with their Phase I and Phase II optimizations. You really can't just apply these glowing tags in any random order.
So there's a strict sequence.
Highly strict. For example, the marker for B cells, which is known as CD20, is incredibly fragile. If it goes through too many boiling cycles, it degrades completely. So it has to be tagged first.
Okay, so tag the fragile stuff before the heat destroys it. Makes perfect sense. But what about the other end of the spectrum?
Right, conversely, some cellular targets are actually masked deeply within the tissue. They require those severe, repeated heat cycles to unfold and become visible. So those absolutely have to go last.
You know, I was looking at how they assign the actual dyes to these markers, and it reminded me of an audio mixer. If you have a really quiet singer and a loud backing band, you don't give the singer a weak mic. You turn their volume way up.
Yeah, that is a highly accurate way to visualize it.
So pairing low abundance targets with the highest brightness dyes prevents them from getting drowned out.
Exactly. It prevents crosstalk, which is when the system gets confused and mistakes one cell for another. By tuning the chemistry this way, they ensure the optimal signal intensity for every single marker is locked firmly between 20 and 25 counts.
So we've solved the chemistry problem. The tissue is perfectly stained, perfectly balanced, and photographed at this blazing 20x magnification. But here's where it gets really interesting to me. How does the computer read all these overlapping colors without just choking?
Well, it uses an algorithmic hierarchy. Rather than trying to process every glowing dot at once, the software follows a strict decision tree.
Oh, so it filters things out in steps.
Right. First, it isolates a marker called CK to identify and exclude all the tumor cells. It gets the bulky stuff out of the way. Then it steps down to look for the CD3 marker, which identifies all your general T cells.
And only after that does it drill down into the specific, rare immune subsets? Why does that specific order matter so much?
If we connect this to the bigger picture, this exact sequence prevents algorithmic occlusion.
Algorithmic occlusion, so like bright markers hiding the dim ones.
Yes, if you don't filter out the bright abundant cells first, they literally hide the dim rare ones from the software. Because they structured it this way, researchers discovered a rare double positive T-cell subset completely invisible to old single stain methods.
Wow. So what does this all mean for you listening? It means this robust blueprint takes highly finicky chemistry and turns it into an absolute standard. It paves the way for next-generation clinical immunotherapy by finally giving doctors a crystal-clear, microscopic map of the battlefield.
And this raises an important question for the future of medicine. If this algorithmic mapping can reveal completely hidden, invisible immune cells in colorectal cancer tissue today, what entirely new microscopic battlegrounds might we discover in other diseases tomorrow?